This protocol is a cell-based method for the in vitro determination of cholesterol efflux in serum or plasma, particularly in high-throughput screenings. This may be applied to diagnose cardiovascular risk and to identify or evaluate cholesterol-lowering agents.
This technique avoids the risks and burdens associated with handling radioactive materials, while providing results with comparable quality levels to the ones obtained in radioassays.
Cholesterol efflux has been reported to associate with atherosclerosis. Thus, the plasma or serum cholesterol efflux of a subject can be compared to a reference control and may provide the risk to suffer from atherosclerosis.
This method could also be applied to other cell lines, like murine cell lines, other human cell lines, or even primary macrophages derived from tissue-resident macrophages or monocytes and human-induced pluripotent stem cells.
To begin this procedure, dissolve the NBD-cholesterol in pure ethanol to obtain a stock solution with a final concentration of 2 micromolar. Dilute 25 microliters of the NBD-cholesterol stock in R10 medium to reach a final concentration of 5 micromolar. Culture THP1 cells in R10 medium at 37 degrees Celsius with 5% carbon dioxide. Every 3 days, adjust the cell density to 300,000 cells per milliliter. Then, obtain a 96 well plate with a flat, clear bottom. For better homogenization, prepare 10 milliliters of THP1 cells in a 15 milliliter tube and add 200 microliters of PMA stock. Mix gently.